Okay, well, while we wait for the slides to come up, thanks for having me here. This will be a fantastic meeting. It's getting off to a good start. And I think most of you know me, but just as a little bit of background, most of my work is on integrating large-scale genome and transcriptome data sets. And I do a lot of work on eQTL mapping. I'm involved in GTEx, other consortia doing this type of work. But today I was asked to talk about the topic of penetrance. And specifically about how we have been trying to understand penetrance by looking at integrated effects of coding and regulatory variants. So this probably doesn't need a lot of introduction to this crowd, but basically a key problem in precision medicine and genetics in general is that even when we have these very, very simple cases of genetics, the sort of like the Mendelian traits or other simple traits where it doesn't get more simple than that, we often have a situation that even when there is a very, very strong genetic association, even as the same variant within the same family, we often have a situation where individuals end up with very different severity of the disease, different types of symptoms. Some people may be completely healthy, some people extremely ill, and others somewhere in between. And this is what incomplete penetrance or variable expressivity means. I personally use penetrance to refer both to penetrance and expressivity. And this is obviously a problem for precision medicine. Our ability to predict phenotype from genotype is hampered by this. It's also a very fundamental biological problem, understanding how do we get from genotype to phenotype. Modified penetrance kind of breaks this connection even for very simple diseases, and thus understanding modified penetrance can give us insights into how this works in general. So us being specialists in analyzing regulatory variants, we explored this hypothesis that is illustrated here, where we basically have a situation where if you imagine a gene where heterozygous loss-of-function coding variants typically cause disease. You may have a situation where an individual, like the one here shown on top, has a cis-regulatory allele that leads to higher expression of the healthy haplotype of that gene, leading to a higher dosage of functional protein product and a better phenotypic outcome than in the opposite case where that loss-of-function coding variant is paired with a lower expressing regulatory haplotype, leading to a lower protein dosage and higher penetrance. And this hypothesis has been explored a little bit previously in some of the earlier work by myself and others in Manolis's lab, single locus papers, some model organism papers. But we decided to take sort of like a big human genetics and genomics approach to crack this. And this work was published a couple of months ago in Nature Genetics and was led by a postdoc in my lab, Stefan Castel. So in this paper, we looked at 3 different types of sort of orthogonal evidence into this hypothesis. First, looking into the general population to basically see, is this even a thing? Do we see any signs of haplotype combinations being sort of changed in the general population, indicating that in the general, more or less healthy population, you have basically decreased penetrance of likely deleterious coding variants. Then we looked at disease cohorts to basically look for the opposite pattern. And then we also did some experimental validation in a single gene by CRISPR. I'm not going to talk about that today. You can see the paper if you're interested. So to kind of like walk you through a couple of the key results from those analyses. First, looking into the general population, we of course turn to GTEx, as we often do in my lab, a data set that we know extremely well. So one of the first approaches how we analyzed this in GTEx was basically asking that when we have rare heterozygous coding variants, do we see signs in allele-specific expression data, looking at the RNA sequencing data from these same individuals, of those variants being more often on the lower expressed regulatory haplotype, indicating that in the general population they are sort of like able to hide there a little bit. And that is indeed what we see looking at the data, comparing rare pathogenic or predicted pathogenic missense variants to other control type of variants. So that's all nice and great. The problem is that, of course, in most human genetic data sets, we don't have RNA sequencing data like we do in CRISPR. So we need to be able to analyze this phenomenon looking at genetic data alone. And the way we do this is that we can basically take just genetic data, take rare coding variants, common regulatory variants, EQTLs from GTEx, and phase those, and basically look whether those rare coding variants are randomly distributed on the EQTL haplotypes, or if there is an enrichment of specific patterns. When we have a pattern where rare pathogenic coding variants tend to be paired with a higher expressing EQTL haplotype, that will be indicating that there is a pattern of potentially decreased penetrance of those variants, and vice versa. So looking into these patterns in GTEx, doing phasing using Illumina reads, both from genome and RNA sequencing data, we see that there is this kind of like a significant decreased penetrance pattern enrichment for rare predicted pathogenic variants, not for controls, in genes that are associated to phenotypes that have particularly strong EQTLs and that seem to be loss of function intolerant, so kind of dosage sensitive genes. So that sort of makes sense and gives us a kind of general evidence that in more or less healthy individuals, there are patterns that indicate that these types of regulatory modifiers of rare coding variant penetrance, that this is actually happening. But of course, the key analysis here was the analysis of disease cohorts, asking whether patients have an enrichment of these kind of like predicted high penetrance combinations of low expression of the healthy haplotype that is alone responsible for trying to maintain gene function. And we looked into two data sets that were available to us. One was the Simon Simplex cohort autism data, comparing the autism probans with their healthy siblings. And then we also looked at cancer patients in TCGA, comparing them to GTEx. And we were here interested in rare damaging inherited or germline variants in disease-relevant genes, a few hundred genes for each cohort or disease, and then and then looking at their haplotypes with EQTLs lifted over from GTEx. And in general, so it was mostly practical reasons that made us choose autism and cancer to analyze. These were large-scale data sets where the genetic architecture seems to be such that rare coding variants seem to matter. We know some of the genes that are involved in this, and we were able to access the data. But this, in general, should work in other phenotypes too. We'll see how well this pans out in further studies. Basically what we did was a comparison of these kinds of regulatory encoding haplotype patterns in these two cohorts, and looking at what these patterns look like for cases and controls. And there's a couple of different things going on here, but I want to draw your attention to these two lines that are the most significant and also the most relevant. So looking at rare variants that are unique to cases. So basically those rare variants that are most likely to contribute to disease risk in cases, we see an enrichment in cases where they are more often paired with a lower expressing regulatory haplotype, indicating that there indeed seems to be a regulatory modifier, common regulatory modifier of disease risk. And then looking at rare variants that are shared both with cases and controls, looking at their haplotype patterns in controls actually shows the opposite signal, indicating that some of these disease-causing variants that are present in controls are actually, these people are controls because of those regulatory modifiers that are basically buffering the effects of those heterozygous loss of function variants. So in general, what we kind of like have coming from these analyses is an integration of two different types of genetic variants that are typically classically analyzed by two very, very different communities using very different technologies and methods. So there is the rare coding variant community looking at large effects in Mendelian and other sort of like severe disease, but mainly by exome sequencing. Then there is the kind of common complex complex disease community, GWAS, EQTLs, association mapping, etc. And we are actually putting these two things together into a joint framework that is motivated by a biological hypothesis of joint effects on gene dosage. And I think that going forward, this type of thinking of basically, not dividing the community by the location of your variant, the frequency of your variant, or the specific disease architecture is going to be really, really important moving forward, both for the rare disease and common disease community. And this is actually something that we are a little bit able to do, thinking about the mechanisms. Some of the work that we published last year has basically allowed us to measure EQTL effects, not just as summary stats of EQTLs, but actual effects on gene dosage. And this allows us to put kind of EQTLs into the same into the same metrics how we think about loss of function coding variants, where we, for example, can see that loss of function heterozygotes obviously lose half of their gene dosage, and this is actually similar to what you get when you have very, very strong regulatory variants. And thinking a little bit about what are some of the lessons that we learned, what are the things that enabled us to do this, and where are we going next? So one of the key things in our work, thinking about genetic modifiers, was that we were actually leveraging on kind of priors from previous large EQTL data sets, looking at genetic variants that we already knew are affecting this target gene of interest, instead of doing this kind of like exhaustive search across the entire genome. So that type of sort of use of priors from previous large-scale studies is, I think, really important and productive without getting into the kind of candidate gene hypothesis kind of issues. As I mentioned before, this kind of combination of biological hypothesis and big accessible disease data sets was something that was essential for being able to do this work. And even though we were able to look into the disease effects and find some significant signals looking at genetic data alone, RNA sequencing data from these cohorts would be even more powerful for detecting these patterns. The data indicates this, and of course that kind of data set would have other uses as well in addition to analysis of penetrance. We are currently working on extending this model and these analyses to additional diseases, additional variant types, looking at splicing variants, etc., different genetic architectures, thinking about how this may apply to even more common diseases than the ones that we studied. One of the things that I want to point out is that this hypothesis that we explored here is really focusing on loss of function variants of dosage-sensitive genes, where basically what matters is the dosage of the healthy non-mutant haplotype. If you are thinking about gain of function variants, actually that flips over, and then the dosage that would matter would be the mutant allele dosage, and that will be an interesting thing to analyze going forward. And one of the things that I think is important to think about in general, and this is kind of a little bit what the discussion was about before, thinking about the gene dosage fitness curve, which is really kind of what our model here is barking at. You could think about sort of an additive model where you just simply kind of the more functional gene product you have, the better it is for your fitness or your phenotype. Recessive variants would be something like that, where the heterozygotes are fine, but then your kind of phenotype value drips very, very steeply once you get to the very, very low dosages of functional gene. Haploinsufficient genes might look like something like this, where there are some areas where even very small changes in gene dosage may actually matter for the phenotype quite a lot. But I'm completely making these things up. I have no idea what these curves look like for different genes. And I think that is actually an important challenge going forward. We can think about various different ways how we can get to this, but both in terms of understanding genes' effect on cellular and higher level phenotypic function, and thinking about interventions, being able to understand how functional gene dosage relates to disease function is important. I'll just acknowledge many people. This is sort of a general list of collaborators. People who are underlined were involved in the paper or are involved in the ongoing work on the penetrance ideas. I'll finish with that.
Say there?
I have a really quick question. So I think this is really interesting. Is there a correlation between dosage sensitivity and human constraint?
Yes. So, well, I guess dosage sensitivity is often discovered by looking at human constraint. Metrics like PLI are really looking at human constraint in some ways. So I guess there are also other methods, but yeah, I think in general that does hold true.
So if you map it out, like it's a linear function, or how does it work?
Oh, I don't think we know if it's a linear function or how like what that curve looks like exactly.
Okay. Manolis and then
So Tuuli, first of all, great talk. You spoke about those loss of function variants in the cohort that are either high in cases or high in controls when you're looking at the EQTL phasing. What about the ones that have equal frequency? Did you see a bias? Because the expectation is that those, basically, this is kind of an effect where it's completely balanced and buffered by the EQTL, and there's no reason to have a difference in frequency because the EQTL is driving the differential effect between case and control.
Yeah, so in this particular analysis, we do the analysis such that we are only testing the haplotype combination, and no differences in variant frequency will affect this particular analysis. However, we've also done the analysis of seeing whether, I think we did this only in autism, but seeing whether autism probands are enriched for having the lower expressing EQTL allele, regardless of their coding variant status. And we do see a signal there overall, basically in decreased dosage in likely autism contributing genes, even from those small effect common variants, seems to matter a little bit.
quick one from Paul.
To what extent do these analyses say anything about lack of additivity or non-additivity? Although as I asked my question, I realized the observations may be too few to answer this at all.
Yeah, very good question. We don't know. So the way we do the analysis, it could be additive, it could be sort of like true kind of epistatic interaction. But yeah, we would capture both, and I think it's a very important and interesting question going forward, how this works. Although I think that I don't think it necessarily matters as much as we sometimes think it does. The sort of like classical epistasis analysis arguing whether these are multiplicative, truly interacting effects is like, here it's like, I mean, it's a modifier. So it's kind of, it's I think, important and interesting per se. And I kind of think that it gets down to this question.